The Victorian era was one of the most dangerous periods in the history of medicine. Doctors were discovering new treatments, new surgical techniques, and entirely new ways to understand the human body. But in many cases, they were experimenting without fully understanding the risks. They didn't yet understand bacteria, they didn't understand blood types, and many of the medical safety rules we take for granted today simply didn't exist. Some of these experiments helped build modern medicine. Others became some of the strangest medical stories ever recorded. Because when Victorian doctors believed they had discovered something important, they often tested it immediately. Sometimes on patients, sometimes on themselves, and sometimes with consequences nobody expected. Here are 3 shocking medical experiments from the Victorian era. What if human blood wasn't the only blood doctors could use? Today, the idea sounds absurd, but in the 19th century, many doctors believed blood itself carried powerful properties. Some even believed personality traits could travel through blood. Calm animals might transfer calmness. Strong animals might transfer strength. So some doctors began asking a strange question. What would happen if animal blood was transfused into a human patient? At the time, doctors knew that severe blood loss could kill quickly, and transfusion seemed like a possible life-saving treatment. But there was one major problem. No one yet understood blood types, and no one understood how the immune system reacts to foreign blood. So in several experimental procedures, doctors transfused blood from animals directly into human patients. Sheep were a common choice. Some physicians believed lamb's blood was especially gentle and pure. In other experiments, blood from calves or goats was used as well. The procedure itself was surprisingly simple. A tube connected the animal to the patient. Blood flowed through the tube and directly into the human bloodstream. At first, some doctors believed the results looked promising, but the reactions soon became impossible to ignore. Patients developed high fevers, some experienced intense pain, others went into shock. In many cases, the body rejected the animal blood almost immediately. Today, we understand why. Animal blood cells are very different from human blood cells. The immune system recognizes them as foreign invaders, and the reaction can be catastrophic. But Victorian doctors didn't know that yet. To them, these early experiments were part of learning how blood worked. Everything changed in the early 20th century. In 1901, Austrian scientist Karl Landsteiner discovered the human blood group system. For the first time, doctors understood that even human blood must be carefully matched before transfusion. Once blood groups were discovered, animal transfusions quickly disappeared from medical practice. But Victorian doctors were still searching for another solution to one of medicine's greatest problems: pain. Before modern anesthesia, surgery was brutal. Patients were usually fully conscious. They had to be physically restrained during operations, and surgeons worked as quickly as possible to reduce the suffering. Speed was not just impressive, it was necessary. So when a new chemical called chloroform was discovered in the 1840s, doctors became extremely interested. When inhaled, chloroform caused rapid unconsciousness. Some doctors believed it might allow patients to sleep through surgery. But there was a problem. No one knew how safe it was. The chemical had never been properly tested on humans. So some doctors decided to test it the only way they could: on themselves. Physicians began inhaling chloroform vapors to observe its effects. The results seemed almost miraculous. Within seconds, people would lose consciousness, pain disappeared, muscles relaxed. For the first time in history, surgery might become painless. But the experiments also revealed serious dangers. Too much chloroform could slow breathing. In some cases, it could stop the heart entirely. Even experienced doctors sometimes misjudged the dose. Despite the risks, chloroform quickly became popular.
In fact, it became so widely accepted that Queen Victoria herself used chloroform during childbirth in 1853. That royal approval helped normalize anesthesia throughout Britain. But later research revealed how dangerous chloroform could be. Many deaths during surgery were eventually linked to the drug. Over time, safer anesthetics replaced it, yet these early experiments proved something revolutionary. Pain during surgery could be controlled, and that discovery would transform medicine forever. But even with anesthesia, Victorian surgery still faced another terrifying problem: infection. And nowhere was that danger more obvious than in early brain surgery. For centuries, doctors believed that certain mental illnesses or strange behaviors might be caused by pressure inside the skull. The idea wasn't entirely new. In fact, the practice of drilling holes into the skull, known as trepanation, had existed for thousands of years. But during the Victorian era, surgeons began attempting more systematic brain procedures. They believed some conditions might be treated by relieving pressure inside the skull. So, in some cases, surgeons drilled small openings directly into a patient's head. The goal was not to operate on the brain itself. Instead, the opening allowed pressure to escape. Sometimes it was used to treat head injuries. Other times it was used in the hope of treating neurological disorders. But surgeons had a major limitation. Without modern imaging, they had almost no way to see what was happening inside the skull. They could only rely on symptoms: severe headaches, confusion, seizures, changes in behavior. Sometimes the procedure actually helped. Relieving pressure inside the skull could save a patient's life after serious head trauma. But the risks were enormous. Sterile surgical techniques were not yet widely used. Bacteria were poorly understood, and infection was one of the greatest threats facing any surgical patient. Many early brain surgery patients survived the operation itself, but died days later from infection. Everything began to change in the late 19th century. Surgeons such as Joseph Lister introduced antiseptic methods. By carefully cleaning wounds and surgical tools, doctors dramatically reduced infections. Suddenly, surgery became far safer. Procedures that once seemed impossible slowly became routine, and the foundations of modern neurosurgery were finally being built. The Victorian era was a time of bold medical experimentation. Some ideas were strange, some were dangerous, and some failed completely. But even failed experiments helped doctors understand the human body better. They revealed the dangers of infection, the importance of anesthesia, and the need for careful scientific testing. Many of the medical rules that protect patients today were written because of lessons learned during that era. Medicine advanced rapidly in the 19th century, but often it advanced through trial and error, and sometimes through experiments that still shock us today.